Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning to everyone who attend this session. I would like to first welcome Associate Professor Daisuke Tsugama from the University of Tokyo. Welcome, Sensei.
はい。Good morning, everyone. So it should be early morning now, right?
I will introduce you. I'll actually make a very brief CV of you, if you don't mind. I will show to the participants. So thank you for your kind availability to share the lecture for everyone today. And so before we are starting the session, I would like to share a brief CV of our speaker of today. Yeah, I hope everyone can see my slide. So I didn't confirm the CV to Tsugama since I hope everything is okay. So today we are going to have the lecture, and the title is the use of reporter genes and recombinant proteins, the technical steps and consideration. And our speaker is the Associate Professor Daisuke Tsugama from the University of Tokyo, Japan. And this lecture is part of a course in the plant breeding and biotechnology study program in IPB University, where I am the coordinator. So for anyone who don't know me yet, my name is Sintho Wahyuning Ardie, and I lecturer in the IPB University. So our speaker today is graduated fully from the University of Tokyo, Japan, from his M.Si until his PhD program, and he finishes his PhD in 2013. So soon after his graduation from the University of Tokyo, he continued his research in University of Tokyo as a GSPS research fellow from 2013 until 2015 in the Graduate School of Agriculture and Life Sciences. And in between of that period, he also became a visiting scholar in biology in the Pennsylvania State of University in the US. And he returned to Japan to join the Hokkaido University as an assistant professor in 2015 until 2018. So after that, until now, Tsugama Sensei, that's how I call him, is an associate professor in the University of Tokyo, Japan, in Asian Natural Environmental Science Center. So you can see his list of publication. You can just scan the QR code showing in this slide, or go to his ORCID profile showing in this slide as well. So that would be the brief introduction of Tsugama Sensei. And without further ado, we would like to invite Tsugama Sensei to deliver his lecture. Please, Tsugama Sensei.
Okay. Thank you, Shinto-san, for giving me this important opportunity. And thank you, everyone, for joining us that early morning. So in Japan, it's 10 o'clock already. So and your time, in your time, it's 8, right? So thank you very much. So I will start sharing my screen. So I can start, right? Probably I cannot share my screen now.
Oh, really? Let me check my security. So sorry. Now, please try again.
Yeah. Is this visible?
Yes, we can see it.
Okay. Okay. So I will start my talk. The title is Use of Reporter Genes and Recombinant Proteins, the Technical Steps and Consideration. And I'm Daisuke Tsugama, and thank you again, Sintho-san, for introducing me. It's perfect. Or too much introduction about me. で、I double checked the meanings of reporter genes and recombinant proteins for this lecture, and I found that recombinant DNA protein has a broader meaning than I thought. I'm sorry for that, actually. And reporter DNA protein is included in the recombinant DNA protein, or it's a part of it. So the title may be confusing, and I'm sorry for that. Yeah. で、はい。ということで、then, I'll move on to the contents of this presentation. So, the, main topics are three. So, the first one is introduction, and the second one is reporter gene protein, and the third one is tagged gene and protein. So, so I will begin with the first part, introduction. The recombinant reporter and tagged gene proteins are like this. So, as I said in the beginning, so the recombinant DNA protein, it has the broadest meaning in these categories of genes and proteins. And the concept is like this. So, recombinant DNA protein is DNA of protein generated by methods of genetic recombination, such as cloning, that create sequences that do not exist in nature. As part of it, reporter gene and protein is existing, and tagged gene protein also exists, and they partially overlap with each other. So in this talk, I'm focusing on reporter and tagged gene protein. Okay, so I'd like to clarify this one first. And I've been working for like longer than 10 years for Arabidopsis thaliana, the model plant. And I was using reporter gene and tagged gene protein a lot for that study. But I'm not introducing my own study actually very much in this talk. So if by any chance have got interested in my study, please check my papers later. I'm only partially introducing my study, and I'm just introducing some general points in this talk. Okay. So these are the points to consider when we use the tagged protein or reporter protein. I think the purpose of the experiment with the recombinant DNA protein is pretty much important always. And for that purpose, pitch expression system, so in vitro bacteria, yeast, insect cells, plants, etc., is used. Is important. For these purposes and expression system, the pitch vector and the features are important. The important thing with to consider, pitch vector and features are used. This is an example of a binary vector, or the vector for bacteria and plants. So this vector is basically for plant transformation, and for that, agrobacterium is often used. And that's why the vector is designed for both bacteria and plants. And this is kind of typical vector for plant transformation. And the point is that looking at this map, we think about the features of the vector. Okay, for this one, promoter is important. I'm sorry. So these are the cassettes for expressing the gene of interest. And in this case, the gene is important, of course, and the promoter choice is also important. And this determines how the gene is expressed in the plants. And in some cases, we want to use reporter gene like this. And how to fuse them or how to use it is important also. In some cases, another gene of interest is put like this in the vector. And for that gene also, promoters and in some cases, reporter is important. For the binary vector system, we will have an antibiotic resistance gene for plants and antibiotic resistance gene for bacteria. These can be together, but these can be separate. These are typical binary vector system. I'm not introducing all the vector system, but the point is that we look at the features and try to understand which features are which, have which function. LB and RB should be the TDN borders. So if I look at this map, I would think that this region, from top to the right, this region will be introduced into plants, and this region, the left bottom region, will not be introduced into plants. So this is just for bacteria, and this is not for plants. Here is another example. This is a PBT32 vector. This is an example of a vector for protein expression in bacteria. So this is a kind of most difficult example, I think, and that's why I showed this one. And I was using this vector when I was a student, and I got several problems in understanding these features, and I got a practical problem in expressing the protein of my interest. So these are the questions relevant to those problems. Looking at this map, we can see that LAC-I gene is present like this here. So what is this gene for? Another problem I had was that I used the XBA1 site for cloning the gene of my interest, and I cloned it successfully, but I failed to express that protein. So in that case, what was the problem? So this is the second question is that one, and the third question is what should be done for the 3' end of the gene to be cloned if it is fused with the histag codons. The important feature of this vector is that this vector can fuse the histag to the C-terminal end of the protein of interest, or histag codons are fused to the 3' end of the gene of interest. So if we want to fuse those histag to the gene of interest, what should we do? This is the third question. And can any of you answer these questions right now? I'm interested. I'd like to give you some time to think about it. If there is no person who can answer now, like 3 minutes. Is there any? If you can answer, then please raise your hand. Don't be shy, please. Sintho さんはわかる?
So, yeah, I think some of the participants are not familiar yet with the expression factor.
Okay. Okay. Then I will give you answers and I will explain the expression factors using this map. Thank you. That information will be very much helpful. So please say, Shinto-san, please help me. If the explanation is too difficult, please help me and I will try to elaborate. Okay. So, please think, like, is it too difficult already? but just think 3 minutes or 4 minutes. Okay. You can also ask some questions regarding the answer. Not direct questions. About other features or something like that.
Bapak Ibu silahkan kalau ini sebetulnya ada pertanyaan yang disampaikan di slide, tapi kalau memang ada yang belum dipahami mengenai fitur dasar dari gene expression factor, silahkan kalau mau ditanya. Tadi ada promoter, ada gene of interest, ada terminator. Apa saja yang bingung dari slide ini silahkan dikonfirmasi ke Tsugama Sensei.
Actually, other features are important to answer these questions.
So I think they are too shy to respond. I'm so sorry.
That's what I thought.
I think there's one student raising hand. Safira, please unmute yourself.
Okay.
Good morning, everyone. Mrs. Ardie and Tsugama Sensei. My name is Safira Rahma from Actually, I'm not from the study program of this session right now. I'm from agriculture. I'm from agronomy and horticulture. So all this our lesson today is a bit foreign to me. I'm sorry. Excuse me. Would you took me back to the previous slide?
Previous. Okay.
Oh well, actually, I took some notes from the about the definition of recombinant protein. So among them is reporter gene. Maybe I'm not. Among them is among the recombinant protein is the reporter gene right now. From in the slide is this green, this with the green arrow. Tsugama Sensei, does that mean the reporter gene is always work around this area? I mean between the gene of interest and the terminator, and if yes, what are its function? Is it to strengthen the expression or anything else? Thank you.
まず、the reporter gene is not always between the gene of interest and terminator. The reporter gene can be used alone without the gene of interest. I mean, the reporter gene itself can be the gene of interest. For example, GFP can be used alone with promoter of interest without this kind of gene.
Oh.
あとは、なんだっけ。Could you say that again? Could you say the second question again? I'm sorry. なんだっけ。
I mean, well, このあたりでファンクションをしたならこのあたりでファンクションをするとはそのファンクションは何者ですか？フンクション What are the function of this reporter gene?
Various. でも、reporter gene is reporter gene, so it reports anything. For example, in the case of GFP, it can be the reporter of subcellular localization of some protein of interest. And so I will introduce GAF gene later, and GAF gene can be the reporter of tissue specificity or stage specificity of gene expression. promoter activity. So if we fuse some gene promoter to gas gene and do gas staining, then we can see the specificity of the promoter activity by the pattern of the staining. And another example of reporter gene is luciferase. If we fuse the promoter of interest to luciferase and express that chimeric gene to promote the luciferase with other effector constructs, and then if the luciferase activity is enhanced by the effector, that means that the effector protein and that promoter upstream of the luciferase are interacting with each other. So the reporters are like that. They report anything regarding the biological processes.
Oh, so it's similar to, I mean, I'm sorry for interrupting. So it's similar to like transcription factor or something?
Yeah, some reporter genes are used for transcription factors, and but Sintho-san is wondering, and transcription reporter gene is not always transcription factor. The transcription factor is often used in this position, gene of interest, and it is fused to reporter gene often. Well, it is the transcription factor gene is used as an effector for the reporter. Effector is different from reporter.
Ah.
I will introduce those example later. It's okay to interrupt like this, though. I'm pretty happy because I'm very concerned about the shortage of, of the content for the length of this lecture. So,
Oh well, maybe the other can ask more question than me. So thank you for the thank you for the explanation. I'm so green about this.
Yeah, but I'm happy if you can interact and ask some questions.
Thank you, Safira. So, Tsugama Sensei, can I add just a very basic information for the other participants? Like Safira is not from our study program, so she has no any basic about biotechnology, molecular biology. Yeah, so maybe a very simple example is the reporter genes may express particular color. For example, GFP showing green color, the gas protein showing blue color. And then if we fuse these reporter genes with our gene of interest, then we can see, for example, where the protein is located. Will it be in the plasma membrane, in the nucleus, or in the other part of the cell, or further in the organ level, maybe where is the gene is expressed, in leaf, in the stem, during flower development. So that kind of reporting function of reporter gene will be discussed in this lecture. So, Tsugama Sensei, I'll return to you.
Some following slides include such examples, but I think I should have included more like colorful ones. I'm sorry, mine is not very much colorful, and I think Wikipedia has more colorful pictures about reporter genes. So please check them also. They are pretty good. So and I didn't use so many. Okay. I will give you the answers to these questions. So first, the LAC-I gene is the repressor for LAC operator. I'm sorry, I have no illustration about that one, but the LAC-I gene is a repressor gene encoding transcription factor that binds to this region in the vector, LAC operator region. And it will inhibit the transcription of the gene of interest. In this, the T7 promoter is a binding site for the T7 RNA polymerase. So, we have to choose appropriate host strain. This is the vector for protein expression in bacteria, and E. coli, Escherichia coli is the most often used bacteria host for this vector. And we have to choose appropriate host for this vector also. the host equalize the Escherichia coli, and the host equalized strain must have T7 RNA polymerase and T7 RNA polymerase. So that kind of specific host cells must be used for this vector. And because BRCA is encoded by this plasmid or vector, so the gene of interest is the transcription of gene of interest is inhibited by this gene, BRCA gene. We can cancel that inhibiting effects by adding some chemical called IPTG or lactose. So IPTG is a lactose analog, and it is not degraded in the cell, so it can last, and it remains effective. So if we add IPTG, then the gene transcription will be turned on. So that's a important thing. And LAC-I, so the answer is that. So LAC-I is used for controlling the gene expression precisely. Very simple is like this. So if I add more, some genes are toxic to cells and it inhibit the cell growth. And in that case, we cannot get a lot of proteins of interest because it's toxic. In that case, we want to replace it first until the cells grown enough in a liquid culture. And if after getting a lot of cells, then we turned on the gene transcription and get the gene product very much. Then we are happy, right? So, and the LAC-I is to realize it, to make it come true. We found this. The second one, if XBA1 is used to clone a gene of interest, the protein from it will not be obtained. Why? It's because if we use XBA1 site, then this region will be removed. ARC-BRES region will be removed. And that's why the gene expression cannot be obtained. ARC-BRES is a ribosomal binding site. Shine-Dalgarno sequence. Do you know Shine-Dalgarno sequence? It's kind of regulatory sequence for prokaryotic gene expression or translation. Protein translation. And this site is AG rich sequence and bound by ribosome. So this is necessary for the for the transcript to be translated into protein. If we use XBA1, this region will be removed. From XBA1 to any of these distribution site will be removed. So ARC-BRES will also be removed, and that's why the protein of interest cannot be obtained. When I did it, I found it, and I put the ARC-BRES back by synthesizing the double-strand DNA with this sequence, AG-rich sequence, into XBA1 site. I just inserted it, and then the protein expression was obtained actually. So that further experiment confirmed that ARC-BRES is pretty important in expression system. And I strongly felt that I needed to understand the feature first before I made that kind of mistake or detour. The third question is what should be done for the 3' end of the gene to be cloned if it is fused to histone. I think this is kind of difficult, but the thing we need to do is to delete stop codon of the gene of interest and make the codon of the gene in frame to histone. In frame. で、if there is one or two insertion between extra bases between the gene of interest, the codon of the gene of interest and the codon of his tag, then the his tag will not work. It will become different codon from his tag, histidine, and it will not translate it as his tag. So, the stop codon removal and codon optimization or the making it in frame is necessary. So this is the answer. I think this is pretty difficult. Actually, I was thinking about the difficult question, and I'm afraid this is very difficult. So, if you are interested, please ask me later. If we have time, you can also directly ask me during this session, and you can also send me an email, and I will try to elaborate. Okay. Anyway, the understanding the features is important. The next is, I hope, the colorful part, reporter gene and protein. The reporter genes are genes that are used to report any biological processes. So how can they do so? And I think I was already giving some answers. So color is one way, and another way is chemiluminescence, and the third one is fluorescence. I think these are almost all as the indicators by the reporter. And are you able to answer what are these? I think color is okay, but chemiluminescence and fluorescence can be confused. And fluorescence can be included in chemiluminescence probably. If we use fluorescence in a, if we use chemiluminescence in a broader meaning. But I usually distinguish chemiluminescence and fluorescence from each other. So, are you able to answer it? Chai?
Ada yang mau mencoba menjawab apa yang dipahami mengenai itu apa perbedaan warna chemiluminescence dan fluorescence dalam hal reporter genes? I'm transferring your question in bahasa. Mas Rizky, please.
Okay, thank you, Ms. Sintho. I'd like to remember about what I got before, but a little bit confused. Not really confused, but I don't know, like not really confident to say that. But I wanna say it for the first question that by color. So like Ms. Sintho said about using the GUST or using the GFP, some of the gene that we add into the plasma membrane will be bright into the different color. So the scientists will understand, will be, will know that the gene is really added into the membrane cell. And the chemiluminescence is using the chemical, something like chemical to extract to make a mark or something like I don't know exactly about this one, about the part 2, and also the part 3 by fluorescence is using UV or something like a light to to make the report genes produce some color or something like that. I don't know. So please give me some. Thank you so much. I'm so sorry before.
Okay. And I think it's almost correct. But I will also explain about color. The color is generated by absorption of certain light, certain wavelengths of light. So we can see the light of the wavelengths from like 360 nanometer to like 650 nanometer. It may be not correct, but it's something like that. We are seeing some very narrow bands of light. Light has so many wavelengths, but we are seeing those lights as the color. で、なんだろう、we can feel the color because some of the wavelengths of color, sorry, some of the wavelengths light will be absorbed by anything. As a result, we are seeing the reflected light, and it is the color. Okay. で、what we are seeing in this case, is not emitting any light. The light source is emitting light, and colored, anything colored, is absorbing that light. So, that stuff is not generating color. So in this slide, we can see black thing, and これはライトだ。これはライトだから違う。ダメだ。not a good example. But, まあいいや。それで、anyway, color is something like that. The light reflected by any object. And the absorbed light is in a certain range. The chemiluminescence is a light emitted by some substance. It is obtained when very often when any chemical is converted by another and as an extra energy. So when some chemical is converted to another chemical, so extra energy will be generated, and it can be a light. It's a chemiluminescence. In that terms, fluorescence is similar. But fluorescence is like in a narrower meaning. So it's a chemiluminescence generated by light. Some light, certain wavelengths of light can excite anything. It will, excite the substance like GFP. And when GFP, the excited GFP, is, go back to the initial state, so the energy will be generated and emitted, and so it is observed as light, and that's a fluorescence. で、more easily, for example, in GFP, we use blue light to excite it, and we see green light as a result. It's a fluorescence. Use some light and get some light. For chemiluminescence, we will not use light to see the resulting light. We add some chemical without light and get some light from the reaction, chemical reaction. So often catalyzed by any protein. That's actually reporter protein. So probably it's difficult to understand without directly using them, but chemiluminescence and fluorescence can be distinguished. So something like that. Okay. Okay. Does that make sense? Or is it too difficult? I think it's difficult to understand. When I was a student, it was difficult to understand. So, okay. Can I move on? Okay.
Yes, please. I put some notes of your explanation in the chat. So later, please review the text, whether it's correct or not, just to help to the participants to catch the information.
Okay, thank you. These are the examples of reporter gene, and gas is one example. And this is beta glucuronidase gene, And so this is kind of enzyme. So the second one is look or luciferase. And this is also enzyme. And this generates chemiluminescence. The third one is GFP, the green fluorescent protein, and its variants. GFP is interesting and useful. I love it. And GFP is also. And other FP genes, FP is fluorescent proteins. I think GFP is the most often used one as FP, but other FPs are present, and they are also used as reporter genes, IP genes, and other reporter genes are also present. For these, I will explain one by one. I'm sorry for character. Gas gene is a bacterial, gas gene is present in bacteria and also human, but for plant study, the bacterial gas gene, UEDA, is often used. We can choose substrates for this UEDA gene product and choose the way to detect its activity or report activity. The first example is X-group. I cannot pronounce this one, but too long. But this is often used substrate, and this is when this is converted to products by the gas gene, so that product includes that product includes water insoluble blue substances, and this is used gas staining. If this is converted to that products, that is insoluble. That's why it can stay in a certain specific region in the cell or tissue or organ. And so that's why it can stay there. If it's soluble, it will move and it cannot be used for visualizing the activity of promoters. The second one is nitrophenyl beta D glucuronide. And the products of this one include water soluble yellow substances. So this is soluble one, and this is used to for the parametric method for reporter assay, effector reporter assay. I will not give the example of it, but color dependent method. This is used for color dependent evaluation of the promoter activity, not in living cells. The third one is MUG, and this is the product from this one includes fluorescent substances, chemicals. So this is fluorescent substance, and this is more sensitive than this one. So lower activity of, for example, promoter can be detected by this fluorescent substances than by this one, parametric method. This is also soluble, so just used for report type assay, something like that. Here is an example, and this is easy to understand, right? I hope so. In this example, these are from Wikipedia for gas reporter, and I was also doing gas staining by myself for the gene of my interest, but this was better, so I'm using this one. And in this experiment, researchers fused the rice embryo specific promoter to gas gene and introduced this construct into rice and subjected its seed for gas staining. And as a result, we can see blue color present in the embryo region. Okay. This is also rice. The right one is also rice. And in this example, rice allele layer specific promoter is fused to gas gene and introduced into rice as a left one. And we can see that embryo has no little color. A little color, but little color, not so much color as the left one. And instead, we can see the blue color in the surrounding region, allele layer region. So, in the way like this, we can visualize in which region in the tissue or organ the promoter is active. In this example, the promoter was already suggested to the promoter was already known to be specific in those cell types, and the experiments are kind of confirmatory, I think. But this kind of experiments can be done for the promoter with unknown activity also. And in that way, we can see if we perform the gas staining with that kind of promoter, we can see the specificity of the promoter, the activity of that promoter. Right? Makes sense? Probably not. But this is kind of most often used as the use of gas in plants. Okay. So here is another example of gas use. So this is the reporter effector experiments. And first we need to, for this experiment, we need to first prepare these constructs. So here are three constructs. Reporter construct includes the Ardie-29B gas. The Ardie-29B gas is one generated by fusing the Ardie-29B gene promoter to the gas gene. The Ardie-29B promoter has ABRE cassette, and ABRE is an ABA response element. So Ardie-29B promoter has a lot of ABA response element or abscisic acid response element. That promoter is fused to Tata box and TATA box and then to gas gene. And this is the one construct needed for this experiment. And we also need to generate effector construct like this. 35S promoter is a constitutively active promoter for plants, for almost any plants, and omega enhancer for translation efficiency, enhancing translation efficiency. And this is the effector gene, and very often transcription factor, but in this example, not transcription factor. These are used as effector gene or protein, and these are casein kinases. So protein, certain protein kinases were used in this example. And so the other construct needed for this experiment is this internal control construct. And this is also a reporter construct. yield quiz なんだっけな、忘れちゃった。ルシフェレス。kind of ルシフェレス。And sorry, I forgot what E stands for here, but this is luciferase and different from gas. This is a point. So, reporter construct has gas and different reporter gene is used here. And it is fused with 35S promoter, constitutively active promoter. These three kinds of construct will be introduced into some cells together. Co-introduced into some, some cell. In this case, Arabidopsis mesophilo protoplast. So, and so in that case, so these effector and internal control genes will be expressed so because of the 35S promoter activity. And if effector gene can activate or can have something to do with ABRE or RD29B promoter, then the expression of gas will be affected. So that's a theory. And the result is in the panel B. Sintho-san, please.
I'm sorry, I need to confirm my understanding about this construct. So we have three construct here. So we are co-transforming a whole cell with three plasmid, or whether this is one construct with three cassette.
Both ways should be okay. Both ways should be okay. But I think, I'm sorry, I don't remember exactly what was done in this study, but this should be from three plasmids, I think.
Oh.
Three different plasmids were co-transformed into cells, I think.
Thank you.
で、this is a result. The top one is a control. And the vector control should be the one where effector plasmid has no effector gene. In this case, some gas and the x-axis shows the relative activity of gas and or the activity the gas activity divided by the luciferase activity, normalized activity of gas. So this is used as a control to normalize how many cells are transformed in that experiment. So we can regard this one as gas activity basically. X-axis as the gas activity. And without the effector, certain level of gas was observed. And in the presence of abscisic acid, the gas activity was enhanced. And that's because of the presence of ABRE acid. This is the abscisic acid response element found by group AB zip transcription factors activating and they activate the expression of promoter activity of RD29B promoter. So that's why in the presence of ABA, gas activity was enhanced. And the point is this. So in the presence of these gene or protein in the effector construct, so we can see these, the top four, top two, CK2 alpha 1 and CK2 alpha 2. So in the presence of them, we can see that gas activity was enhanced, even in the absence of ABA, and in the presence of ABA also, compared to the top one control. We can see that the third and fourth effector were not effective, not effective as the top two effectors. And we can also see that the bottom four effectors are all repressing the activity of this promoter or gas activity, right? So even in the presence of ABA, so the activity of gas was very low. So when these four effector genes were present. So from this result, we can see that CK2 alpha 1 and CK2 alpha 2 are promoter kind of activators of the gas activity or RD29B promoter activity, at least in this system. And these CK2 beta 1, CK2 beta 2, CK2 beta 3, and CK2 beta 4 are the negative regulator of ARD29B promoter or ABR gas activity. So, this is called reporter assay or reporter effect assay, and gas can also be used for this kind of assay. So in this case, the protoplasts were broken, and their soup was used for the experiment. Right. The next is luciferase. The luciferase is enzyme that can generate light by converting some chemicals. And these are the relationship between the luciferase and their substrates, chemiluminescent substrates. The firefly luciferase will use luciferin as a substrate. And Renida reniformis luciferase and their relatives are using soeren terazine. ソエレンテラージン。違うかな。ソエレンテラージン。うん。ソエレンテラージン。で、these are all marine organisms. And I think this kind, I don't like the shape of these organisms, but they are marine ones. And some are pretty, I hope. And echorin is from, なんだっけ、クラゲってなんていうんだっけ、クラゲなんだけど、this is also from, this is also living in the sea, and this is also using soeren terazine. で、あと、bacteria also has luciferase, and its substance, substrate is lumazine. で、anyway, and these substrates are different from each other. So if we use different kinds of luciferase, we can change the substrate, and it is sometimes useful. So here is an example of luciferase reporter assay. So in the previous example, for reporter to construct, gas was used, but in this case, luciferase was used. The effector in this case was BR worky 65, and this is the transcription factor from brassica rapa. Chinese cabbage かな白菜 or Chinese cabbage, and it is fused to 35S promoter. And so these people wanted to see whether this transcription factor can activate the promoters of interest like this. In this example, in the reporter construct, there is another expression cassette, and it is 35S promoter with Renila luciferase. Renila luciferase is this one, and this luciferase should be firefly luciferase. So that's why we can distinguish the signal from my baby's back. From the school now. Sorry. We can distinguish the signal from Renila luciferase from firefly luciferase because if we change the substrate. So that's why this can work.
So just to confirm, so the use of the REN controlled by the 35S promoter is like a control.
Yes.
To be compared with the luciferase expression later. Is that so?
They the most important value they wanted to obtain should be the firefly luciferase activity. As you said, the Renila luciferase was used as the for the normalization of the
normalization. Okay, understood. Thank you.
Yeah, and they were as in the previous example, introduced into some cells. Tobacco, as I know, like tobacco. maybe wrong, but anyway, they were introduced together in cell, sorry, and after some incubation, the fire-applied luciferase was normalized by vanilla luciferase and expressed like this. So we can see that in the presence of this WRT65 gene or protein as the effector, so all the promoter activity was enhanced. Right? That should be right. So, that suggests that this transcription factor interacted with these promoters and enhanced the firefly luciferase activity in this experiment. Here is another example using luciferase. In this example, RB29A gene promoter was fused to luciferase and introduced into Arabidopsis plants. And after introducing this gene, so the plant was made homozygous for this transgene and put into some mutagenizing dangerous chemical, dipped into chemical, and mutation was induced, and the resulting seeds were sown in the stress plates, and their luciferase signals were observed under unstressed and stressed condition. In this case, cold stress was given to them. These are plants. Is it understandable that these are plants? These are Arabidopsis seedlings, very small plants. And so from B to D, the signals are showing the luciferase signals, light emitted by these plants. In theory, all of them should have luciferase gene like this. It's difficult to see, but under unstressed condition, almost all the plants have no signal. But here, the highlighted one, it has signal, and this is called the cos mutant. In D1 and D2, cold stress was given to the plants, and after that, the luciferase activity was detected like this. And this is a longer exposure version, and this is a shorter exposure version. And in the longer exposure version, signals are generally strong because of the long exposure. But there is a plant that was generating low signal, weak signal, and this was called LOS mutant. Even so, RB29 is a stress responsive gene, and its expression is enhanced by multiple stresses like drought is a good one, but also cold stress and ABA treatment and salt stress. And this is kind of expected result. A lot of plants are generating a lot of luciferase activity, but this plant was not doing so, and so that's why they thought that this is mutant. In this example, the exposure period is shorter, and even under this condition, so one plant was showing strong signal, and this was called FOS mutant. The cos mutant is this one, and this is constitutively this has constitutive stress response, right? Even under unstressed condition, so it was showing the signal. And LOS mutant is low stress response mutant. Even under the stressed condition, it was showing the low luciferase activity. host is, has high stress response. And that's because it was showing the strong signal like this, stronger signal than the other plants under the stress condition. So, I'm afraid this may be difficult to see the difference between the signal in the signal strengths, but luciferase can be used in this way to screening for mutants that have different responses to environmental stresses or other stimuli. Okay. And this is another example of luciferase we use. So this is a split luciferase for protein-protein interactions. So in this system, we split the luciferase into N-terminal part and C-terminal part, and fuse them to the protein of interest, in this case, X and Y. And if the proteins X and Y interact with each other, the luciferase, N-terminal part and C-terminal part of luciferase will come close to each other. And the luciferase activity will be reconstituted, and then luciferin will be converted to the product, and the light will be emitted. So by this way, we can see whether the protein of interest are interacting with each other. And this is the example. I think this is tobacco, and yeah, the protein of interest was prepared like this. They were interested in the interaction between OSPIC1, so rice-PIC1 protein, and OSXLG2. I'm kind of familiar with this protein, G protein probably, and membrane protein. I think they were interested in this combination of proteins, and this was OSPPI was used as a negative control. The two constructs expressing these X group N and Y group C were prepared for each of these combinations, and those two constructs were infiltrated together in these regions of tobacco leaves. This is a luciferase signal obtained, and we can see that in the left part, so there was no signal, and in the right part, there was signal. This is luciferase signal. This should be right, but expressed as this color chart. So this means that this can be interpreted as a positive interaction between those proteins.
Just to confirm, so in this experiment, we can say that the BIK1 protein is interacting with the SLG2. That is not interacting with the PPI. Am I correct?
Yes, that's correct. This result can be interpreted like that. Because the luciferase activity was obtained, the protein should be interacting with each other.
So in this experiment, is a transient expression?
Yes, I think this is transient.
Thank you.
Two proteins of interest were expressed by agrobacteria, I think, infiltrating the agrobacteria cells into tobacco leaf.
Okay.
Yeah.
So it's not transforming the plants, the whole plants.
But not split luciferase, but I'm talking about the BFC later. And for BFC, stable transgenic plants are sometimes generated for some specific purpose. So that may be true for this experiment as well. I've never seen such an example, but some people may have been doing so. And the panel B is a quantification for the panel A, and the panel C is a confirmation of the presence of those proteins in these leaves by Western blotting. And if we use reporter gene or any protein tag, Western blotting can be easily performed compared to the Western blotting using specific antibody for a protein. I mean, if we want to directly detect the protein X and protein Y, we need to prepare two kinds of antibody specific to those two proteins. And raising antibody can take time and cost. So, if we use tag, we can just buy some commercial antibodies for those tags and reporters. Because those reporters and tags are often used, so commercial antibodies are often available for them. And it can be cheaper and more useful for doing Western blotting. That's another thing using those tags and reporters. Anyway, split luciferase is kind of becoming popular, I feel, as a protein-protein interaction assay. So next is GFP and FP genes and others. GFP and its derivatives are interesting. So I think GFP is FP isolated from coerurescence by Osamu Shimomura. And Shimomura sensei is a Japanese person. And this the Shimomura sensei was the discoverer of the GFP. Or Shimomura sensei is the person who first isolated the GFP from this organism. We call it Krage, and I forgot how to call it in English, and I'm sorry for that. Jellyfish, jellyfish, it's jellyfish. And Shimomura sensei went to get a lot of jellyfish with his family, and that story was good for me, interesting to me. So I thought it nice to get a lot of jellyfish by using the family power. He isolated GFP from jellyfish. The good thing about GFP is that it's small, 27 kD, and hydrophilic and stable. Easy to handle. The original version of GFP is excited by UV, but later many scientists got interested in GFP, and a lot of variants have been made to improve fluorescence spectra and the brightness of GFP. So, GFP is good. These are the GFP derivatives. I hope you have ever, maybe not true, but from the original version of GFP, YFP, PFP, and BFP were generated. And I showed them with the color. So this is a GFP variant. And these mutations were introduced into the original version of the GFP. And these mutations are to make the excitation light from UV to blue light and enhance the brightness. So this was a very important mutation. And this made it possible to express the GFP stably in human cells, animal cells. And because of this one, certain the subcellular localization of certain protein was detected by GFP, and those guys got the Nobel Prize with Shimomura sensei. I think 3 people got the Nobel Prize because of GFP, and one was Shimomura sensei as a discoverer, and the other 2 were the people who utilized the GFP well for the research. So, and EGFP mutation was a kind of key mutation for those later studies. And YFP are the GFP variants that have yellow, near yellow color. Actually, the color is like green to my eyes, even if it's called YFP. But it is called YFP. And these are the mutations for them. And the mutations are close to the fluorophore, the region that is responsible for making the color for the fluorescence. Fluorophore. So it's relevant to the mutation sites are relevant to the fluorophore positions. The CFP is a cyan FP, cyan fluorescent protein, and they are also used by these mutations. The citrin and cerulean, for them are bright versions of the YFP and CFP respectively. The BFP is a most blue FP from the original version of GFP, and this was made by this one. Okay. The other FPs are also used, and red FP variants isolated from this one, corals, also popular as a reporter gene. So the most popular one is probably DS red, so the red fluorescent protein. I don't remember the species name exactly, but DS is a species name, and it's also coral related species. And the M.Si is a variant of DS red. The GFP and DS red has to have different sequences, but their structures are similar. So it's interesting, I think. Beta barrel, it is called beta barrel structure to get the light and to emit the light. Many other variants with different excitation emission spectra were generated from different species. So it is diversifying. So FPs are diversifying, and I think it's useful and interesting. And those diversified FP include photoactivated, photoconvertible, and fluorescent timer, etc. Photoactivated FP is a FP that becomes active only after getting some light. So they are not fluorescent in a steady state, but once we expose them to certain light, then they will be activated. And this is the photoactivated FPs. And the photoconvertible is FPs that emit fluorescence, 2 kinds of fluorescence. In the steady state, it is emitting specific fluorescence. And once we expose them to a specific light, then they will emit different fluorescence from the original one. Like yellow one, first yellow one, and then red one, something like that. Fluorescent timers are like short time, short lifetime fluorescent proteins. And as a reporter, this is kind of good. They become fluorescent, and then they will die. So they can be stable only for a short time. Let's be the example. And this is the example of GFP, colorful one. This is from my paper. I was handling the gene called BIP1. This is from Arabidopsis. And in this example, I expressed BIP1 GFP and POSF21 and ATB29 are homologs of BIP1. I expressed these proteins as GFP fused proteins in Arabidopsis stably, the whole plant of Arabidopsis. And I put the roots of them in water, hypoosmotic solution, and observe the GFP signals. And what I thought interesting is that the GFP signals were first detected in the cytosol, but later it was detected in the nuclei like this. So dots are nuclei in these pictures, and then they were detected back in the cytosol. That phenomenon was not observed in the GFP alone. So that's why I got interested in this one, and I studied a lot for this one. I don't know if I can say I worked hard for them, but I got fond of this one because of this one. Another example is using M.Si and the GFP together. Doing this experiment, in the experiment for the right pictures, sorry. M.Si and GFP were introduced into onion cells together. The bottom one is showing the GFP alone, M.Si alone, and overlay picture. And the top two are showing the GFP fused to a transcription factor gene from asparagus. Do you know asparagus? No, vegetable. AO-35 and AOMS are those transcription factors from asparagus, and they are fused to GFP. And we can see that the only limited region in the cell is green in these examples. In contrast, M.Si can be could be found in the cytosolic regions. So by this kind of example, if the experiment, we can see the subcellular localization, and in this case, nuclear localization of these proteins. For the GFP or IFP, this is kind of a complex experiment, but showing good things for the FP. PFC and FRET for protein-protein interactions are used sometimes. PFC is a bimolecular fluorescence complementation, and FRET is a fluorescence resonance energy transfer. In this figure, the number one should be for PFC. For the PFC, we will prepare split YFP or split GFP like this. Like split recipients. We split the GFP into N-terminal part and C-terminal part. And we will fuse them to the proteins of interest. In this, for example, protein A and protein B, respectively. We will co-introduce them into some cells. And if the protein A and protein B interact with each other, the N-terminal part and the C-terminal part of the GFP will come close to each other, and they will become fluorescent in that case. The split GFP is not fluorescent, and only if they come close to each other, they become fluorescent. That's the theory for PFC. For the thread, the thread is shown in number 3, and the negative interaction is shown in number 2. For thread, we will fuse protein A to, in this example, YFP, and protein C to CFP. This should be CFP, and this should be YFP. If the protein A and protein C interact with each other, these proteins will come close to each other, and some fluorescence from CFP will be transmitted to YFP as the excitation light source. This is a fluorescence resonance energy transfer. In this case, the fluorescence lifetime for the CFP will be decreased. And using some specific microscope, we will detect that decrease in the fluorescence lifetime by the thread, and see. And in that case, the protein of interest will be interacting with each other. If that lifetime is not changed by the protein A and P, then they are not interacting with each other. That kind of negative interaction is shown in the panel 2. Yeah. They are far from each other, and no energy transfer will be happening. And in that case, the lifetime for CFP is unchanged. The pattern 4 is showing the BFC thread. So this is a combination of BFC and thread. In thread, in this example, the CFP is called donor, and the YFP is called acceptor. And in the panel 3, regular YFP was used as acceptor. And in the panel 4, the reconstituted YFP is used as the acceptor. The theory is pretty the same as the panel 3. So if the protein A and B are coming close to each other, the YFP is reconstituted. And if the C can come close to that complex, then CFP to the reconstituted YFP energy transfer will happen. And if we in that state detect the lifetime for CFP, then it should be decreased compared to the original one. The 5 and 6 are showing, the importance of the conformation. If the orientation of the YFP and CFP are not good, thread will not occur, even though the protein A to C are interacting with each other. And it's a case for BFC also. It's a case for BFC also. This kind of situation is possible for regular BFC without CFP. So, the orientation of protein is important. Okay. So this is a example of the result. So, this picture has a lot of information. We should look at this figure like this. This is one cell in one cell. Panel B and C are for one cell. The panel D, E, and F should be for one cell. And the panel G, H, and I should be for one cell. And in this one cell, in the top case, CRFP, HV bump 731 was expressed. This is CFP. CRFP is CFP. And CFP HV bump 721 was expressed in the test example. In the middle example, this protein, the same CFP fused protein was expressed with these proteins. NYFP, HV, ROR2, and HV SNAP34, CYFP. This is for BFC, and this is for CFP. They were trying to show the BFC threat in this experiment. So in the top example, only one protein was expressed. And in the middle and the bottom example, three proteins were expressed together in one cell. The right picture is showing the CFP lifetime, as shown here by color chart. In the bottom example, that CFP fused protein was expressed, and these two proteins were expressed. And the difference between the middle one and the bottom one is that HVSNAP34 orientation of this protein. In the middle example, CYFP was fused to the C terminal of the SNAP34. In the bottom example, the CYFP was fused to the N terminal of the SNAP34. So this is the difference. The difference is only in the orientation or the position of the CYFP tag. All the other conditions are the same between the middle one and the bottom one. Okay. The result of interest is the right one, green, or fluorescent lifetime imaging. We should there are several points of interpreting this. So the first one is that we can see the signals of YFP like this. And this means that BFC is okay in both of these cases. So orientation about these are okay. The YFP part is coming close to each other in both of these orientations. And another thing we can see is that the pattern of the CYFP, CFP fused to protein. Without these two proteins, the CFP fused protein was observed in the cell periphery like this. But in the presence of these two proteins, signals are like this, aggregated. So this is another evidence that these three proteins are interacting with each other. The most important one is right one. We can see that this color is not as blue, is not so blue as this one, right? The fluorescent lifetime is longer if the color is very blue. And if it's shifted to red like this, then the lifetime is shorter. And we can see that the middle one, in the middle one, the CFP lifetime is shorter than the bottom one or the top one. This means that threat is happening here, and no threat is happening in the bottom. So in this case, the middle one is showing that the orientation is right, and it is further showing that those three proteins are forming the complex. They are close to each other. Even without the threat, probably some interactions can be suggested by this result, this change of the localization of this CFP, this protein, and the BFC. But this is kind of typical result of BFC threat. Good one. I think it was complicated, but で、other reporter proteins include these, beta-galactosidase, laxi. This is a good one. Sintho is smiling. And I like this one too. Very simple one. E. coli, for E. coli or bacteria. And we can use colorimetric method to pick up the colony of interest, right? And for cloning, it's very useful for cloning. Good one. Alkaline phosphatase is also regarded as reporter protein. We usually use some colorimetric method to make blue color generating substance or chemiluminescent substance for substrate for alkaline phosphatase. The peroxidase is also like that. Like alkaline phosphatase, it is used for detecting some as a reporter using blue color generating substrate or chemiluminescence generating substrate. Sorry. Yeah. So, these are not as colorful as FP, and not so many examples as FP. So, just introduce. The last part. Protein tags are like this. GST and MVP, his tag, small tags, and others. And I will explain about them one by one also. First, GST, MVP, and his tag. GST, sorry. The GST is glutathione S transferase. So MVP is maltose binding protein. His tag is hexahistidine, and it's kind of variants. The small tags include flag, HA, MIC, and other ones, and others. GST, MVP, and his tag. GST is 27 kD soluble protein, and it binds reduced glutathione. This kind of ligand or the small molecule interacting with these tags is kind of important. I'm introducing the affinity chromatography dependent protein purification later. Basically, this interaction is for purifying them by affinity chromatography. MVP is maltose binding protein and 43 kD, it's larger, a little larger than GST, but it's soluble. The maltose and amylose binding activity is had by them. His tag is 1 kD, or the molecular weight of histidine is like 155. So if we multiply 6 to it, so it's like 0.9, and 950, 900. So it's like 1 kD, and molecular mass is 1 kD, and histidine is water soluble, but we can, if this is used with protein, we can induce aggregate often. So it's kind of problematic. But his tag is also useful anyway. So because it binds metals such as nickel and cobalt, heavy metals. Heavy metal dependent chromatography is we will call it IMAC, immobilized metal affinity chromatography. Not computer. The his tag is anyway used for that IMAC. And the flag HA, MIC, and others. The flag tag is DYK, DDDDK. This is all. So it's very small, so consisting of 8 amino acids. And DDDK is enterokinase cut site. So if we fuse this tag to protein of interest, we can cut this tag by enterokinase. And this is sometimes useful. And HA is hemagglutinin tag from some animal virus. And this is also small, YPYBV, PBYA. And the MIC tag is also from virus, and this is also small, EQKLISHAAEEPL. Another example of small tag is strep tag, WSHPQFEK. And the strep tag can interact with strep, the protein called streptavidin or streptavidin. And that interaction can be useful from affinity chromatography as well. And other examples of tags are S tag, PRX tag, etc. I think this is difficult to remember, but anyway, some large tags are used and some small tags are used, and they have pros and cons. ラジャtags are more likely to inhibit the protein, more likely to inhibit the function of the protein of interest if they are fused to them, generally. And the smaller tags are thought to be less inhibiting the functions of the protein of interest. Okay. This is showing the affinity chromatography of GST. We can buy the agarose labeled with GSH. GSH is a reduced glutathione. And first we equilibrate this agarose with GSH, and we then add the protein solution with GST fused proteins. And because GST has affinity to GSH, so the GST fused protein is bound to that resin or agarose. And the other proteins should be whether if they are not interacting with that GST fused proteins, they will be gone. So only GST fused proteins will remain on the agarose, and then we will wash them to further remove these contaminants, and later we will elute these proteins by competition by adding the reduced glutathione, and then we will get these protein solution as a purified protein solution. Okay. This solution is useful for many protein experiments. Here I'm showing the result from the protein purification. These are showing this is showing the Western blot result after the purification, and this is just showing the presence of these proteins. And this is the position for GST alone, and this is the signal very strong. And because they are chimeric protein, so the size is larger than GST alone, they are detected like this. And for GST-BG52 and GST-BG30, the positions of those signals are almost the same as the GST alone. So probably there is no fused protein. So the expression was failing in this case. I very often use the purified protein for the further protein experiments. Not very often. I mean, when I do the protein purification, the purpose is usually for the protein assay like this. And this is the example, in vitro phosphorylation assay with the purified protein. In this example, these upper two, top two panels are just to confirm the presence of the purified proteins in the purified protein solutions. So I used the purified protein solutions for Western blotting and just detected the protein of interest like this. In this case, GSTB1 and MBPCPK21. And in the bottom, so I mixed these proteins with ATP and calcium and incubated them and detected them by the phosphorylated protein specific probe, FOSTAG. And I could detect the MBP-CPK21 specific signals and the GSTB1 specific signals using that FOSTAG. And so, in this case, BIP1 can be regarded as phosphorylated by the MBP-CPK21. They are both purified form, so it is good. Because it can exclude the possibility that other contaminating proteins, phosphorylated to BIP1 or something like that. So using purified proteins is good in this kind of experiment. Another example is gel shift assay to evaluate the DNA binding ability of the transcription factors or purified proteins. In this example, GSTB1 and GSTB2, these proteins were used. These proteins were the same as one here. So I did this experiment to confirm the protein purification and then used them for gel shift assay like this. Here, the detected one is probe DNA if 707, 707 A1 promoter. They were mixed with proteins and electrophoresis, and their signals were detected, and DNA signals were detected by chemoelimination method. And the point is that in the presence of the protein of interest, the band will be shifted. So in the presence of GST alone, the probe is detected in this position. And in the presence of BIP1 and its homologs, the band was detected like this in the upper positions. So this can be regarded as a result of interaction between these BIP1 and its homologs and DNA, CYP707 promoter in this case. This protein purification can be extended to study the interaction between those proteins and the small molecules, small or large molecules. This is showing red. So in the purification process, this can be in the first example, the GSH was used, but for MVP, amylose is used. Nickel is used for his tag, and antibody is used for some tags. If we use antibody for purification, it is called the immunoprecipitation experiment, IP or immunoprecipitation. But the experiment handling is pretty much similar between immunoprecipitation and the other protein purification by affinity chromatography. Okay. We will then use appropriate tag for according to these reagents. And if these protein of interest has interactors like X and Y, like this, then even after washing, they can remain on the protein of interest. In that case, these substances X and Y will be eluted together with the protein of interest like this in the final step, or elution step, after the elution step. These can be protein, DNA, RNA, and I forgot to write, but small molecules are also included, like calcium or heavy metals or whatever. By any way, we can detect these molecules, and then we can say that these molecules are interacting with the protein of interest. So, many protein, many interaction assays are possible for the protein and the molecules, but some of them are using pretty similar theory for detecting the interacting molecules. Okay. I'm just introducing some example and wrap up this talk. So this is an example of protein-protein interaction study using those protein purification ways. So it is for the in vitro pull-down assay. And in this experiment, so GST BIP1 was immobilized like this. So, on the agarose first. And then, so it was later reacted with a purified form of his tagged ATB prime delta protein. After reacting them, so the protein was washed. Basically, GST-BIP1 was washed and eluted. And the presence of his tagged protein was assessed. So there should be, of course, GST-BIP1 because it is pulled down. And the interest was in the presence of his ATP prime delta. If they interact, if this protein interacts with BIP1, it should be present in the elutant. And it should, if the ATP prime delta does not interact with BIP1, it is not present in the elutant. So it is a hypothesis, and the result was that the ATP prime delta, his tagged ATP prime delta was interacting with BIP1 because the signal was detected like this. And the right panel in this figure is pretty much similar to the left one. Sorry, but in this example, truncated versions of BIP1 was used. N means that N terminal part of BIP1 and C means C terminal part of BIP1. So the right panel was done to narrow down the regions in the BIP1 interacting with the ATP prime delta. Sorry, pass is a homolog of ATP prime delta. And the interact because the signal was obtained only when GST-BIP1C was used. So this means that the C-terminal part of BIP1 is interacting with pass, and the N-terminal part of BIP1 is not interacting with pass so much as C-terminal part. I showed this one. It was a design for experiment and what the gene of interest means. This is another example of protein purification mediated examination of DNA protein interaction. The experiment is called chromatin immunoprecipitation or CHIP. And in this example, chromatin immunoprecipitation was performed for the BIP1 GFP. First, BIP1 GFP was purified by antibody. Because antibody was used to purify it, it is called immunoprecipitation. And after that, DNA was eluted from the precipitated BIP1 GFP. And the presence of CYP707A1 promoter and CYP707A3 promoter was checked by the PCR. And this is showing that the antibody used is shown here, and the BIP1 GFP, the presence of BIP1 GFP is shown here. For minus, I used the GFP alone. The whole BIP1 GFP plus, I used the BIP1 GFP expressing plants. We can see that only when GFP antibody was used to purify BIP1 GFP, the signal of these promoter fragments were obtained. This means that BIP1 GFP and these promoter fragments are interacting with each other. And immunoprecipitation is regarded as in vivo experiments. So, this is kind of good. This experiment is difficult, but good to say that interaction is in vivo. So,はい。で、I think the last part was difficult, but this is summary, very short summary. Important thing is which vector and features to use. And reporter genes, proteins, and tag genes, proteins are useful. It has many uses. So, the thinking about the purpose and about the purpose is important, and choosing them is important. So, I'm sorry, but I think this is very difficult. But this is all from me. So thank you very much for listening.
Thank you very much, Tsugama Sensei, for the lecture. It's very important that you are introducing gene reporting and also tag. For me, tag is new because I don't use tag before. So thank you very much. So so the participants now, if you have any question, we still have some times. I put some summary also in the chat for easier information to you to grab. But if you need something to confirm, please just open your mic and ask your question.
これはすぐに答えられない。
Any
I'm afraid answering the questions on the chat will take time, so I think.
No, I don't put any question on the chat. The chat only contains my notes for the participants.
これは
Only the summary. So we have one participant raising hand.
はいはい、お願いします。
Yes, please.
Yeah, thank you.
Matsutsuki first.
Oh yeah, thank you, Ms. Sintho, and thank you for Sensei Tsugama that bring me so many knowledge that improving my understanding about the importance of reporter gene too. And the question is, I'm so sorry if the question is really basic because it has been a long time that keep in touch with biotechnology. I guess almost 2 years probably. Okay, the question is about all of the reported genes that you said to us. Which one the most accurate or the most economist or something like the most useful for general purposes? I'm sorry.
I think it's GFP.
Okay. GFP. Yeah. So the most simple sequence here, GFP with the that you say to me that this is really easy to use. So GFP is really common to use it, Sensei.
FP is also useful.
Oh, okay. And also I want to ask you about the, I'm sorry, about the effector gene. Effector gene that you say before, this may enhance the result of GAS. So it is just a specific gene for enhancing the GAS expression, or probably we can use for another purposes like to increase the gene expression or targeted gene expression, or just specific for GAS.
In this in this experiment, we can regard the data as like 概念だからちょっと難しいんだけど、the point is that the point is promoter, the upstream the gas gene, this promoter was used, Ardie29B promoter or ABRE sequences. Because of this gene, this was activated, and that's a point. You can use any kind of this any kind of gene for this effector position. Actually, this is not very typical pattern. These are protein kinases, and very typical pattern is transcription factor. So some putative transcription factor gene is inserted here. And to discuss whether they can bind to this region and affect the expression of this gas reporter gene. But in this case, protein kinase was used, and protein kinase affected the ABA, the abscisic acid dependent pathway, and that's why this kind of result was obtained. This is not very typical, but this is interesting, I think. Because of those protein kinases, the downstream transcription factors should have been affected, and that's why this promoter was activated in the presence of these, and by the presence of these. Does this answer your question?
Probably once, if I have a time.
Yes, please. But just to clarify the first and the second question, just to confirm also my understanding here. So the effector genes affecting the promoter region, and then the effect of the effector genes in the particular promoter, in this case is the RD29B or the ABRE cassette, is reported by the signal of gas. So your question was whether the effector gene is specific for gas, but the explanation from Tsugama Sensei is that the effector genes affect the promoters that controlling the reporter genes expression. In this case, is gas. Am I? Please confirm.
Thank you.
It is kind of interesting viewpoint. Nobody should think in that way. Effector may have some direct effects on gas activity, but it is never discussed. We just usually discuss the effects on promoters. So it may be possible that cousin kinases can phosphorylate the gas gene, gas protein, and affect its activity. Some direct assessment is necessary to verify that, but it is possible. Now I think it's possible. Kind of interesting.
So
thank you so much.
Your third question, if you have.
Okay, probably a simple question, probably about the time of or the duration of the reported genes that you said before. I don't get the most important thing about that, like using the something like a fluorescence timer or something like that. So how we use that method for general purposes or the specific purposes, or something like what the most benefit to use to lengthen the fluorescence or the reported genes activities duration.
Is your question specific to fluorescent timer?
Yeah, timer. Yeah.
Timer はね、actually, I have I know no good example. I'm sorry for that. But the lifetime for the reporter protein is important, I would say. Luciferase has shorter lifetime. The GFP has generally long because of its stability. It's hydrophilic and it is stable protein. So it is usually has a longer lifetime. And in some report, and that's why the GFP is not usually use this kind of reporter assay. I didn't introduce the reporter assay with GFP here, right?
All right, yeah.
Because of that stability, GFP is not good for that kind of reporter assay. But if you use a shorter lifetime GFP, like fluorescent timer GFP, then this kind of thing can also be done. And more importantly, probably it can allow more like reporter in vivo. This is the reporter says requiring the breaking the cell. But most important benefit of GFP is that it can be observed in vivo, in situ in vivo, without killing the cells.
Oh,
We just have to give the blue light and get the green light. That's the GFP. And if we use the fluorescent timer, it may be more precise. We may be able to get more precise response to some stimulant or some promoters upstream of those fluorescent timer. That's my guess.
okay, thank you.
I have to survey some examples.
Thank you very much, Sensei. Thank you very much, Ms. Sintho.
Thank you for question.
Thank you, Mas Rizky. So I think we can have one more question here from the participant. I see Ms. Safira, you are raising your hand, so please go with your question.
Okay, thank you for the chance, Mrs. Ardie. From the lecture, it fills me so much with so much information, so much new information. For that, I'm thanking you so much, Tsugama Sensei. From all the recombinant proteins that we know, especially the reporter genes and the tag protein. And from all the examples you've given to us in this lecture, it's all regarding to the it's all related to experiments, like detecting the one proteins or genes in everything related to researches. So about this reporting genes and the tag protein, how can we adjust the activity span of these proteins? So can we do that? There you go. Maybe I can tell this with Indonesian. I'm sorry, Miss Ardie.
Yes, please.
Okay.
I'll try to grab your question in English later on, please.
Oke, jadi bagaimana kita bisa mengatur span waktu kapan si protein reporter atau tag protein ini aktif gitu, aktif seperti itu. Jadi bisakah kita atur, bisakah dimaksudkan ini aktifnya hanya pada saat purpose tertentu, atau ini bisa digunakan pada saat waktu yang lebih lama, atau bahkan bisa bekerja dalam waktu yang selama dia disisipkan dalam organisme tersebut, seperti itu. Terima kasih.
Okay. So I'll try to translate the question from Ms. Safira here. She is asking, how can we control the expression of the reporter genes? For example, like this example, you can control the expression only in particular tissue. And we also mentioned that the stability of some reporter can be detected only in the short time. Some are longer. Ms. Safira asked whether it's possible to have the reporter genes all the time as long as the genes is inside the plants. So I think that was her question. So
no, and I'm a user of the reporter, and some other researchers are trying to generate different versions of the reporter genes. So we users are just dependent on those researchers to be optimizing or diversifying the stability or properties or whatever about the reporter genes. We are just choosing some of them according to our purpose. So does that answer your question? In this kind of example, we will use promoter. In that case, we will use same gas as left and right. In this example, we just use different promoters, and in this case, we can compare the promoter activities between left and right. but we usually don't care about the gas activity in this case. We just use them without thinking so much about the, we usually rely on other researchers. But it may be possible to predict some, point mutation that can unstabilize or stabilize those proteins, gas, and on purpose use the gas variants. But usually it is out of our purpose. So we are interested usually on the promoter activity and not in the gas activity, not tweaking the gas activity. I'm kind of interested in those things, but it's not the main study for us. So, does this answer your question?
Sorry, a helicopter just passed my location, so
okay.
it's a little bit busy. I try to transfer your answer in more general explanation in bahasa Indonesia. Jadi Mbak Safira terkait pertanyaannya mungkin mudah-mudahan saya bisa meneruskan jawaban dari Tsugama Sensei. Mbak Safira sebenarnya pertanyaannya ada dua. Apakah kita bisa mengatur begitu kan kapan si reporter gene ini aktif begitu ya. Nah, kalau pertanyaan pertama itu diaturnya dengan promoter. Jadi kita punya namanya constitutive promoter, maka kalau pakai constitutive promoter seperti 35 CAMV, reporter gene-nya akan muncul terus-menerus di bagian manapun dari sampel kita. Tapi kalau promoternya misalnya stress inducible seperti Ardie29B, maka si gas tadi akan muncul hanya pada saat keadaan stres. Begitu. Nah, jadi itu diaturnya oleh promoter. Tapi kalau pertanyaan kedua, apakah kemudian si protein reporter ini bisa terdeteksi selama gennya ada di dalam tanaman? Nah, itu kaitannya dengan stabilitas dari reporter. Nah, jadi kalau stabilitasnya itu maka yang tadi disebutkan ada lifetime. Ya, berapa lama dia bisa diobservasi. Sebetulnya selama dia diekspresi, nanti tergantung berapa lama dia bisa diobservasi. Mudah-mudahan itu menjawab pertanyaan dari Mbak Safira.
Thank you very much. I'm sorry for resorting to Indonesian.
Yeah, I'm sorry to explain in Bahasa because it's easier to explain in Bahasa. So, do we still have more questions? We are already 10 past 11 here. So I think it's lunchtime in Japan.
It's okay. Are you okay? I'm sorry for taking so long.
So if we still have some questions from the participants, do we have any? Okay, I think we don't have more questions. It can be asked later on for my students in my classroom if you have any further questions. So because we are already 10 past 11 here, we would like to thank you, Tsugama Sensei, for his inspiring lecture. It's very informative, even for me. I didn't use tech before. I knew that Tsugama Sensei did many experiments with GST, and I never understand what is that. So today I know a bit about that. Thank you very much. And I would like to extend the gratitude from our study program, the head of study program, Professor Dewi Sukma. She joined this lecture in the beginning, but she have to left the Zoom because she has another agenda. And also from our department head. So kindly allow me to pass the electronic certificate to show our gratitude for your time and knowledge that you have shared to us for your the use of reporter genes and recombinant proteins in the molecular biology. And this is handed from Professor Edi Santosa, our head of department. He is also the graduate from Tokyo University under the supervision of Tsugama Sensei.
Hi.
So thank you very much. So, and maybe before we close this session, I would like to have a group picture with everyone. Can you please turn on your camera? Okay. We have 2 slides in my screen. So I'll count to 3 for the first screen. If you are ready, whenever you are ready. Okay. If you can't turn on your camera, please put any reaction. Okay. First screen to go. 1, 2, 3. Smile. A moment. I need to pause here. Okay, and second screen on mine. Okay, 1, 2, 3. Smile. Oh, I have only few faces in the second screen. Okay, and I would like the participants to fill the attendance list because I would ask the department whether it's possible to provide you with a certificate of attendance of your participation today. So Tsugama Sensei, thank you once again. And
Thank you again.
please tell my regards to everyone in the lab, and also thank you for your wife because taking your time this morning.
Thank you. Thank you very much, everyone.
Thank you very much. So kindly allow me to close the Zoom. Bye-bye.
ありがとうございました。
Shoot, stop recording.